Hi, my name is Rhiju. I'm a faculty member at Stanford University School of Medicine, and I lead a citizen science project called Eterna. While you're watching this talk, I invite you to play Eterna at eternagame.org and to take screenshots and tweet about what you're watching at Eternagame. Before starting the talk, I'd like to dedicate our research and this presentation to the many players who have been affected by COVID-19, and specifically to Malcolm Watson and to other players who have passed away as a result of this dreadful pandemic. The COVID-19 pandemic and all the health inequities that it is causing and all the deaths that it is causing will not end until two events happen. First, a vaccine needs to be developed and authorized for medical use. And second, that vaccine has to be injected into everybody in the world. A key towards ending the pandemic may be a new RNA technology. And you may have heard about mRNA vaccines. These were the first molecules that were injected into human beings as potential vaccine candidates. And I'm showing you a picture of that first injection that happened in Seattle with this patient named Jennifer Haller. There's now nearly 20 mRNA vaccine efforts underway around the world. And it does seem like by fall of this year, at least one and maybe several of these molecules may be licensed for use as emergency COVID-19 vaccines. But there's going to be a potential problem. Okay, so RNA vaccines are bottlenecked if you're going to try to get them into everybody in the world. You see, once an RNA molecule is inside saline solution and in a syringe ready to inject into a human being, it starts to decay on the timescale of hours or days. And this poor stability, even under refrigerated conditions, means that it's currently difficult to manufacture and distribute RNA vaccines in pre-filled syringes in the same way that you or I get our flu shots. And this poor stability is a property, a biochemical property, that's intrinsic to RNA molecules themselves. Starting last year, before the pandemic, some industry folks had contacted me in my lab about potentially solving the mRNA stability problem by redesigning RNA molecules so that they might code for the same proteins, but fold up into these tightly packaged structures like what I'm showing you here in the bottom. So there's evidence in the literature suggesting that if you could do this, the RNAs would remain stable in vitro for significantly longer than standard mRNAs, and their potency as vaccines would be sustained and perhaps even enhanced by this RNA structure. The problem is we don't have design rules, much less algorithms, to carry out this kind of design of highly structured RNA vaccines. So I reasoned, this is last year, that it might take another 3 or 5 years to get to this point. We would carry out high throughput experiments, deep learning, and algorithm development to figure out how to stabilize mRNA therapeutics and vaccines. But we don't have 3 to 5 years. We now have 3 to 5 months. How can we accelerate our research by 10x? I know of one way to tackle a currently intractable RNA design problem with 10x the speed of normal academic research, and that's through a citizen science platform called Eterna. Eterna is a video game whose tagline is "Played by humans, scored by nature," and it was launched in 2011. Since that time, we have made available to video game players on the internet a mildly addictive game-like interface that you can play in your web browser, and players return to us sequences of A, C, G, and U that they think might fold up into the target structures or have the target functions that we need for our design challenge. They then publish their solutions and vote on which ones they like best. And then my lab and our colleagues at Stanford take the top-ranked solutions. We actually synthesize them in a wet lab and carry out high-throughput experiments to test their structure and function. And we return these data back to the players. And through this virtuous cycle of gameplay and wet lab feedback, Eterna players have created RNA sensors and robotics that are forming the foundation of the next generation of RNA medicines. They have created new kinds of RNA calculators that underpin a cheap point-of-care diagnostic for TB and they've created technologies that we hope can allow for gene therapies to be turned on and off reversibly for safer deployment inside human bodies. The Eterna community has grown to over 250,000 registered players and has given rise to over 20 scientific publications, including 9 that have involved Eterna players as co-authors. I'm showing you here a paper published in 2016 where the 2 lead authors are Eterna players. This paper was published in Biochemistry a few weeks ago. All of the authors here are citizen scientists. No one from my lab or elsewhere at Stanford is on the paper. And here's my favorite example. This is a preprint that I believe is the most important scientific paper written in RNA design in the last 5 years. I wish I had written it, but instead it was written by a single Eterna player named Fernando Portela, a former white hat hacker who had no prior connection to science and in fact never went to college, and yet has produced this remarkable work that completely outperforms all deep learning methods that my lab and about 15 other labs have created to help solve the RNA design problem. At the beginning of 2020, when the COVID-19 outbreak became pandemic, we realized that we needed to steer this entire Eterna community and research process to stabilizing RNA vaccines in the hopes of enabling worldwide immunization. And that is the goal of the OpenVaccine COVID-19 challenge. To stand up this Open Vaccine challenge, we've had to overcome 4 major kinds of problems that we'd never faced before on Eterna. We had never put out puzzles that try to stabilize mRNAs that code for proteins. And we never looked at an RNA above 1,000 nucleotides in length. But remarkably, several experienced players stepped up and in fact designed these puzzles that let us launch OpenVaccine in March of 2020. We realized that we wanted players who are most affected by COVID-19 to be the ones who are most involved in the research. So we have taken enormous development cycles to try to port the Eterna game from a web browser game to a mobile game, and the native apps are going out this week. We have established partnerships with scientific education programs like the NOVA TV program to get the word out to people who have been stuck at home during the shutdown. And we've established partnerships to get Eterna into communities that have not previously been big on Eterna before. These include international communities through a partnership with King Abdullah University in Saudi Arabia, as well as several partnerships with historically Black colleges and universities, with a very strong partnership with Peer Review A&M just outside Houston. And through these efforts to reach out to new players, we're now getting thousands of potential vaccine solutions every week. Now, of course, we need to test them experimentally. And again, before the pandemic, we had not yet developed, and no one had developed the experimental technologies to test thousands of vaccine candidates for their stability and protein translation efficiency all at once. But my lab, in collaboration with Maria Barna's lab in Stanford Genetics, under waivers from Stanford leadership, have carried out social distance experiments to invent the technologies that we've needed, and we are filing our 3rd patent on these experimental innovations later this week, and are completing our 2nd and 3rd rounds of Eterna experiments for OpenVaccine. And last, of course, we need to take extra steps to get our RNA molecules out into clinics, and we are now in what I hope are the last stages of negotiations for our first partnership with one of the big RNA vaccine industry consortia to take our molecules. If we're successful in stabilizing their RNA vaccines, they have agreed to put them through rapid animal safety trials and then into phase 1 through phase 3 human clinical trials. If all goes well, then Eterna stabilized molecules could have an impact on the pandemic as early as late December of this calendar year. It's been a priority to us to have our research go out as open science, and we've been putting out preprints in May, June. 2 more will be going out in July and August. The first papers are on computational innovations that we have discovered, and the last 2 papers will cover our experimental validations and new methods. And although it sounds like science fiction, if we're successful, then we'll see that the public invention by nearly 100,000 people on this Eterna platform could lead to the RNA medicine that then gets injected into billions of people to end this dreadful COVID-19 pandemic. To end, I'd like to thank many people, a developer team who we've recruited across 8 time zones, a huge team of scientists at Stanford University, including advisors in Po-Su Huang and Hua Chu's lab. In addition to scientists, it turns out lawyers, administrators, IP experts, development officers have all been critical in ensuring that OpenVaccine could be launched without delays. I'm grateful to partners like the PBS NOVA program and Professors Lorenz and Regisford at Prairie View A&M for helping spread the word about OpenVaccine. All of the OpenVaccine research has been funded so far by unrestricted gifts from donors and private foundations. I've listed them here on the right. I'd like to thank our players, our core developer team, as many of you know, our former players or active players. LFP6, Jay, Nicole, and Ome all were recruited from the player community. I'd like to thank a large group of players themselves who have really stepped up to take part in the Open Vaccine challenges. And I'd like to invite all of you to come join us at eternagame.org and help us solve this Open Vaccine challenge. Thank you.